All right. So we will be beginning with Sarah Parr. She's our first presenter. Yes, if you will turn on your cameras, give her your full attention. Thank you, Sarah, for presenting.
Yeah, of course. Let me get my screen shared really quick. Okay, well, hi everyone. I'm Sarah Parr. I am a senior neuroscience major with a biology minor on the pre-med track here at Baylor. And I'm also an intern in the VA Center of Excellence Research Program. It's a internship that is a partnership between Baylor Pre-Health and the Vision 17 Center of Excellence for Research on Returning War Veterans at the VA here in Waco. So all of the interns in this program have to pick a topic to do a literature review over, and we work on conducting a literature search and analyzing articles and coming up with a poster and a paper. So this is the information that came from my poster, and the other authors on my poster were Dr. Zambrano Vasquez and Dr. Kraus, who are the advisors for the CERP internship, and then Dr. Austin, who is my PI. And I chose to talk about the effects of transcranial magnetic stimulation and ketamine infusion for treatment-resistant depression. So when I started my literature search, I was just really interested in treatment-resistant depression and wanted to figure out which treatments I wanted to specifically focus on. And in my search through the literature of treatment-resistant depression itself, I found a lot of staggering statistics, like over 8.4% of adults in the US have experienced or will experience a major depressive episode in their lifetime. And those who meet certain criteria will then be diagnosed with major depressive disorder. And another really shocking fact is that at least 2/3 of death from suicide each year in the US occur while that person is actively in a depressive episode. So unfortunately, within the population of patients with major depressive disorder, it's estimated that about 30% don't achieve remission from first-line antidepressant treatment, which is like the gold standard antidepressant medications like SSRIs, commonly known as like Lexapro, Prozac, other things like Cymbalta, things that are just widely known as antidepressant medications, and also psychotherapy. And so when these patients are not able to achieve remission and they keep experiencing depressive episodes, it can contribute to an increased risk of suicide. So the most widely accepted definition of treatment-resistant depression is the failure to achieve remission from 2 trials of these first-line antidepressant treatments with the proper dosage and duration. So it can be a little bit hard to define because sometimes medications aren't given with the proper dosage and duration, but this is currently the best working definition for TRD. And there's a lot of risk factors and varying causes for TRD, just like there is for depression. Some of them are overlapping and they're the same risk factors or same abnormalities that happen in major depressive disorder, but then some are specific to TRD. But the heterogeneity of cause for depression makes it really difficult to treat all patients. And that's why not every treatment works for every patient. So some current theories suggest that it could be abnormal volume of certain brain areas like the amygdala or the prefrontal cortex, or abnormal connectivity of different networks within the brain that could be causing TRD. But because of the risk that it poses for suicide, for just impaired daily functioning, and also the significant healthcare burden that it creates, it's really important that we find alternative treatments for TRD. It's estimated that TRD versus non-TRD depressed patients spend $2,000 to $3,000 more a year on healthcare. So that's a huge difference in the cost. So the objective of my literature review was to examine the efficacy of IV ketamine infusion, as well as transcranial magnetic stimulation as standalone treatments for TRD, as well as look into the promising effects of combining the two together for a treatment called CTK, for combination TMS and ketamine, to increase treatment success for those who don't achieve remission on monotherapy. So I did a literature search with keywords related to ketamine, TMS, and TRD, and I ended up finding 18 original data articles and 3 review articles that I included in the review for this poster. And I first started with the results of each treatment on its own. And for TMS, the mechanism of why they think it helps depression is it's really just a big magnetic coil that they hold over the skull, and when the magnet is turned on, it induces an electrical current and stimulates the brain area that's below it. And when TMS is used on the medial prefrontal cortex, the front part of your brain, or the parts of the cortex that are just below it, it's been shown to improve depression. And a lot of studies have shown through a review of TMS studies, it creates a response in approximately 40% of TRD patients, which is great because these patients didn't have any response to their previous treatments, but it's still a little bit low, like why only 40% if the mechanism seems to work? And one of the reasons for that is because it's hard to have a high level of treatment adherence to TMS because it takes 20 to 30 treatment sessions to achieve the desired effect and to achieve remission from depression. And there also can be some unpleasant side effects like headache, pain at the site of treatment, some lightheadedness, because it is an intense electrical stimulation. But it does show a lot of promise for treating major depressive disorder. It's also been implicated in treating other disorders that are typically comorbid or happen along with major depressive disorder, like PTSD and opioid use. Now, ketamine is a very different type of treatment, and it's been really looked into for its rapid effects. So ketamine is a strong antagonist for a receptor in the brain that is receptive to an excitatory neurotransmitter, glutamate. The receptor is called the NMDA receptor. And so ketamine blocks it, and that's its main mechanism of action. And for IV ketamine infusion, they typically will do a dosage based on the person's body weight and infuse it over a span of about 30 minutes. And a single ketamine infusion has been shown to improve symptoms within 2 to 4 hours and lasting significantly through 3 days. But after about a week, it falls off and it doesn't have as much of an effect, which is a very opposite problem from TMS, where it takes a really long time to see the effects, but then produces long-term effects. So there's also been recent research that has made an intranasal spray form of ketamine, which has improved its accessibility, and it actually has been FDA approved specifically for treatment-resistant depression, which is really interesting. So then there was one lab in particular that was doing a treatment of CTK, combined TMS and ketamine. And some background for why they believe that would work is that ketamine blocks these NMDA receptors, allowing for other receptors to be targeted in the brain, and it alters the excitability of the brain. And that's important because TMS is trying to stimulate it. So if it's easily stimulated, then the effects of TMS could work better. So it alters it to make it more excitable, but in certain areas, it's also shown to just balance the excitability and the inhibition. And there's also ketamine originally is an anesthetic, and so it has some pain relief and some dissociative anesthetic properties, which can lessen the unpleasant side effects of TMS. And this seems to be really efficacious in the treatment groups and the case studies that this lab did. Of all patients received or achieved at least partial remission, most achieving full remission, and most of them were still in remission 2 years post-treatment. They went 3 times a week, and they had anywhere from 10 to 30 sessions. So these are really good results for patients all having remission when none of them responded, once again, to treatments prior to that. And another cool thing was that they saw some other behavioral outcomes other than just the standardized measures of depressive symptoms, like returning to work, relationship success, going back to school, increased rates of marriage, substance abuse recovery, and things like that. So it's really promising that this could help people in their lives overall. So with the difference of TMS taking a long time, having a lot of sessions needed, it's not really ideal because a lot of people will drop out of the treatment before they actually achieve the effects. So they don't achieve the long-term remission. But ketamine can cause reduction of symptoms really quickly, like I said, 2 to 4 hours. So putting the ketamine with the TMS could possibly increase treatment adherence because they'd be seeing improvement sooner. But then augmenting ketamine, which doesn't last very long, with TMS that we know does last a lot longer, could compensate for that lack of long-term effects. So they really do balance each other out. But there are drawbacks for each one. So there's not a whole lot of literature at the moment seeing if there would be any negative side effects. There haven't been any that were found in the treatment groups that I reviewed, but that could be something later on in future research that could be discovered. Like I was saying earlier, the NMDA channels are a low frequency receptor. And when TMS is used, it's given at a high frequency. So if these low frequency channels are blocked, the stimulation and the ketamine can target these higher frequency receptors, and that can help the excitability and help the brain be more receptive to TMS. And if one session is more efficacious because the TMS helps, then that could possibly decrease the number of sessions needed, which helps the problem of treatment adherence. And like I said before, with the pain relieving side effects or effects of ketamine, CTK may be more tolerable than TMS alone to help with the headache and things like that. But this improvement that's been shown in treatment groups so far has been significant and definitely warrants further research in this area. And there's only one lab publishing this treatment so far. So obviously it should be replicated to be able to be generalized to the rest of the depression population. And there are not any placebo randomized controlled studies. So that would also be something that should be looked into in the future. Thank you guys so much.
Thank you, Sarah. That was wonderful. Does anyone have any questions for Sarah? All right, well, I have a question for you. I was wondering, like, what made you get interested in this research and like, I don't know, like what was the beginnings of this and like, what was the initial intrigue, I guess?
Yeah, so at the VA CERT program, I am in the neuroimaging core. So I work closely with a lot of clinical psychologists, neuropsychologists, and so there's obviously a focus on mental health, especially with veterans having PTSD, depression, a lot of times. And so I just started looking into treatment-resistant depression, and I found that TMS was really interesting. One of my other PIs, she is a neuropsychologist that does TMS studies, so that kind of piqued my interest. And the VA is also opening ketamine clinics in a lot of locations. So I just found that they had the most evidence and the most literature as individual treatments, and I happened to cross a lab that combined them. So it was really interesting.
Super cool. Thank you so much for presenting.
Yeah.
All right.
Thank you
so
guys
we're gonna
so
move on. Yeah, of course. We're gonna move on to our next presenter, Abhinav. If you could share your screen and make sure to have me pinned, that'd be wonderful. And we're gonna get started. Thank you so much.
All right, are you able to see my screen? All right, hi everyone, my name is Abhinav Sankaranthi. I'm a junior at Rice University, and I'm an undergraduate researcher at the Venkatachalam lab at UT Health McGovern Medical School. My presentation dives into the spreading patterns of pathological tau in Drosophila larval brains. So what brought us into tau spreading is that a hallmark of neurodegenerative diseases is protein aggregation. Specifically for Alzheimer's disease, it's characterized by memory impairment and cognitive decline resulting from extracellular amyloid beta plaques and intracellular tau filaments. And we wanted to focus on tau's role for accelerating the progression of Alzheimer's, as it is a relatively untapped field that can yield promising new discoveries and findings. Brack staging tells us that the severity of AD, or Alzheimer's disease, is correlated with pathological tau spreading volume in the brain. So how exactly does tau spread? Well, literature tells us that normal tau proteins can become pathological through hyperphosphorylation, leading to the detachment from microtubules and the formation of filament structures when aggregated. These intracellular pathological tau tangles can disrupt healthy neuronal functions and thus contribute to neurodegenerative diseases. We suggest that preventing the spread of tau can be helpful to slow down the progression of Alzheimer's disease. And we find that when we express human tau in Drosophila brains, we notice a spread that could be evolutionarily conserved. We're observing both human and bovine tau in third instar larval brains of Drosophila. Specifically, we know that the tau protein originates in the axons of glutamatergic neurons in the Drosophila brain. And thus, we look for targets that can moderate the amount of tau that is being spread away from these neurons to determine amount of tau spread and correlate that with Alzheimer's disease. So there are two specific targets for our experimentation. One is inositol triphosphate receptor, and the other is transient receptor protein channel for the mucolipin subfamily. So why did we choose these two targets? Well, studies show that calcium dysregulation is linked to neurodegenerative diseases such as Alzheimer's disease. And the inositol triphosphate receptor is characterized by its function to release calcium ions from the endoplasmic reticulum based on various cellular factors. Similarly, tryptophan, or transient receptor protein for the mucolipin subfamily, conducts calcium ions as well, specifically from the lumen of lysosomes to the cytoplasm. And what this shows is that these two receptors are linked to calcium ion regulation, which could then be correlated to Alzheimer's disease. Additionally, previous findings in our lab show that the expression of tau shortens the lifespan of Drosophila, while the hindrance of ITPR signaling can lead to an increase in the lifespan of Drosophila tau mutants. And ITPR, again, is the inositol triphosphate receptor. So a shortened lifespan is a phenotype for Alzheimer's disease-associated genes. So these findings and the fact that these receptors are involved in calcium ion regulation allow us to hypothesize that the knockdown of ITPR and tryptophan can limit tau spread and potentially halt Alzheimer's disease progression. So moving on to our methodology, for this study, we dissected third instar larval brains of various genetic crosses for the Drosophila that we use in our experiment. We then fix these brains in a solution of paraformaldehyde and perform primary and secondary antibody staining. After the antibody staining is complete, to visualize the tau or glutamatergic neurons where the tau originates, we will mount the brains onto a microscope slide and image them with confocal microscopy, where we will run a Z-stack protocol. After we've developed these images, we'll then analyze them for tau volume and the overlap between tau and the glutamatergic neurons. So for the control, ITPR knocked out and tryptophan knocked out groups, the red fluorescence from our antibody staining represents human tau localization, and the green fluorescence represents glutamatergic neuron localization. And we obtain the overlap between these two in order to have a representation as to how much tau has stayed in the glutamatergic neurons where it originates versus how much of it has spread. So for the bovine tau group, which is another one of our experimental groups, the red fluorescence from our antibody staining represents the glutamatergic neurons, while the green fluorescence represents bovine tau. And we perform similar analysis for all groups to determine the overlap of tau and glutamatergic neurons. So for our preliminary results, as you can see in the preliminary results section, we have a bunch of larval brain images Figure A shows the UAS MCDA GFP UAS tau group, which is basically our control group that just expresses tau. Figure B shows the triple knockdown group. Figure C shows the GFP-B tau group, which is bovine tau. And figure D shows the brains of the ITPR knockdown group. And overall, for our experiment, we're going to determine how much tau is localized to the glutamatergic neurons in each group. So spreading, more spreading would be correlated to a lower value of tau localized to these neurons, because tau is is originated in these neurons. So if you have more spreading, you're going to have less of a fraction of tau localized to these glutamatergic neurons. So the results that we obtained for our current trials do not suggest a significant difference between triple IR or triple knockdown and the control group. As you can see in figure F, around 49.72% of tau protein is localized to the glutamatergic neurons in the control group, while there is there's 49.72% of tau protein localized to the glutamatergic neurons, while 45.7% is localized to the glutamatergic neurons for the triple knockdown group. And we noticed that overall, there's around 8% less tau localized to glutamatergic neurons overall for the triple IR group compared to the control group. However, we can't make definitive claims on this trend due to a very high p-value of 0.35, which is much greater than 0.05. Thus, we can't reasonably reject the null hypothesis that there is no significant difference between the two groups. So for our next steps, we plan on continuing more experimentation so that we have a large enough dissection sample size to improve the consistency of our results and data. So we're going to continue more experiments with the control group, ITPR knockdown, triple knockdown, as well as GFP tau. We're also going to identify any inconsistencies in our methodology to figure out why our p-value was so high and why we had some inconsistencies in the data that we got for the triple knockdown group. Furthermore, we're going to continue doing more image analysis for the brains that we have currently dissected and quantify the amount of tau that has spread away from the, or that is localized to the glutamatergic neurons where it originates. We're also going to determine if there is a difference between spreading patterns of bovine versus human tau to find out if this spreading of tau is dependent on type of tau protein. Our more holistic goal is to determine the mechanism behind pathological tau spreading and determine potential targets, mostly through genetic alterations in Drosophila, and find any noticeable patterns and trends of decreased tau spread for these genetic crosses that we can then use for more experimentation and can hopefully lead to the development of Alzheimer's disease therapies. And that's pretty much it for my presentation. Does anyone have any questions?
I have a question, Alvin. First, love your presentation. And second, so my question is, why did you decide to study this specifically in Drosophila as opposed to like rodent models, I guess? Because I'd imagine it's pretty difficult to extract larval brains.
Yeah. So the reason why we work with Drosophila is because of the generation time for Drosophila and the amount of time it takes for Drosophila to grow and to cross them with basically develop these Drosophila crosses, and the time it takes to produce Drosophila larvae, which is the target for my experimentation, is kind of, it's not that too long. So we can develop many crosses and have many vials of these Drosophila that in a week will have Drosophila larvae that we can then dissect. And contrary to, I mean, it sounds like, dissecting a larval brain is pretty difficult, but I think it's not so difficult as long as you know the protocol. We just basically cut the Drosophila larvae open, It's pretty small, so we do that in the microscope slide, or in a microscope, and we then like extract all like the gut and other organs and kind of locate the brain, and then we take it, clean it up, and then uh, put it in a tube and continue to do like fixing as well as antibody staining. And eventually, if we see trends in the data that we get through doing experiments with Drosophila, we will then move on to mice to see if there's a similar effect in mice.
Awesome, thank you.
Does anyone else have any questions? All right. Well, thank you so much, Abhinav, and he came all the way from Rice. So thank you so much for joining us in Baylor. Means a lot. And we will move on to our third presentation. This one is by Grace, Laura, and Riley, and Kayla. So go ahead and share your screen whenever you're ready, and we'll get started. Thank you so much.
Okay, can everyone see the PowerPoint? Okay. Good morning, everyone. My name is Laura, and I'm here with Grace and Riley, and we are representing Baylor University Psychoneuroimmunology, or PNI lab, under Principal Investigator Dr. Vichaya and graduate student advisor Kayla Gill. Today, we will be presenting on exploring the impact of hyperglycemia on cognitive and affective outcomes in mice. So according to the National Diabetes Statistics Report in 2019, 37.3 million people in the United States have diabetes. This is about 1 in 10 people, or 11.3% of the entire US population. In 2019 alone, there were about 1.4 million Americans that were diagnosed with diabetes. Also, the CDC has stated that the percentage of adults with diabetes increases with age, where about 29.2% of those 65 years and older have diabetes. So right now, there are two classified types of diabetes, type 1 and type 2. Type 1 diabetes is characterized by the autoimmune destruction of beta pancreatic cells, which leads to the inability to produce insulin. Whereas type 2 diabetes is characterized by the dysregulation of the body's response to insulin and/or insulin resistance. Both types do lead to hyperglycemia, also known as high blood sugar levels in the blood. This causes an increased risk of a host of other issues, such as peripheral neuropathy, anxiety, cognitive impairments, and/or dementia. So, to follow up with the risk of diabetes, our study explored its cognitive and affective outcomes, as this health condition is often comorbid with depression. In literature that surrounds preclinical studies, there have been results that display type 1 diabetes significantly increasing depressive-like behavior. Additionally, these preclinical diabetic models have shown a progression of cognitive decline, which also includes what we are interested in investigating further. So therefore, it is important to continue to investigate diabetes, more specifically type 1 diabetes, from a neurological perspective. So in this diabetic study, we chemically induced hyperglycemia in mice by utilizing streptozotocin, or STZ, which is an agent known to preferentially destroy beta pancreatic cells. This inhibits the mice's ability to produce insulin and successfully mimicking type 1 diabetes. As we will see throughout our presentation and from prior research, STZ is known to produce both cognitive and affective symptoms associated with diabetes, such as depressive and anxiety-like phenotypes. To further explore this concept, we hypothesized that hyperglycemia would induce significantly heightened anxiety and depressive-like phenotypes, alongside poorer cognitive performance in mice injected with STZ when compared with normal control mice. And to test this hypothesis, we evaluated a diverse set of affective and cognitive behaviors and the STZ murine model to investigate the impacts of hyperglycemia on our diabetic mice.
In the study, we used the C57BL6J mouse strain, which is the most widely used mouse strain in studies of human disease models. We had a total of 13 male mice in cohort 1 for this study who were injected with STZ, or the control citrate buffer, at 7 weeks. We are currently working with cohort 2. The mice were group housed with 2 to 3 mice per cage, all containing mice of the same treatment in the vivarium here at Baylor. At 7 weeks of age, we began treatment. We injected 7 mice with 50 milligrams of STZ for 5 consecutive days, and 6 mice were injected with the control citrate buffer. Post-injection, we monitored their weight and fasting blood glucose consistently throughout the study in order to ensure the health of the mice, as well as to confirm that the STZ mice had become diabetic. The threshold for diabetes is 250 milligrams, and so we wanted to ensure that immediately following STZ injection that we had reached that threshold. At week 20, past injection, we performed a vast variety of behavioral testing on the mice. In week 21, the mice were cold, and we examined their tissue. Next, we will discuss the behavioral data for this study. We found significance in open field, marble bearing, forced swim test, and fear conditioning. So here we see the blood glucose and the body weights of the control mice compared to the STZ mice. We see that the mice became diabetic immediately following the STZ injection, surpassing the diabetic threshold of 250, with our control mice keeping their blood glucose levels well below the line. We also see that the STZ mice consistently weighed less than our buffer mice, increasing their body weights at a slower rate as compared to the controls. However, all the mice were at a healthy weight all throughout the study, with their weight and general health monitored very closely during daily and weekly health checks. So one of the tasks we performed on the mice was open field, which is an anxiety task in which we place the mice in a box and monitor their total distance moved, as well as the amount of time in the center field of the box for 5 minutes. If a mouse is more anxious, we tend to expect to see decreased time spent in the center field. However, in this study, we see a significant decrease in the total distance moved with no significance in amount of time spent in the center field. We believe that this decrease in distance moved is indicative of a depressive-like phenotype in the STZ model. Marble bearing is another anxiety task in which the mice are placed in a cage with bedding and 20 marbles. For 30 minutes, we allow the mice to bury the marbles, and at the end, we determine how many marbles were buried. We typically expect to see anxious mice burying more marbles than the control mice. In our study, we see a significant decrease in marble bearing in STZ mice. Similar to open field, we believe that this is also indicative of a depressive-like phenotype and has been explored further in the literature to confirm this. Next, we performed a forced swim test, which is a depression task in which the mice are placed in water for 6 minutes, and the percent immobility is measured compared to their swimming activity. Immobility is correlated with a lack of motivation and a depressive phenotype. It is important to note, however, that mice are incredibly buoyant and very good swimmers. So the immobility in the mice was just them floating around in the water as compared to swimming. Here we see a significant increase in the amount of time spent immobile for the STZ mice as compared to the control mice, further indicating this depressive phenotype that we've seen in the other tests.
So the last behavioral task that we conducted was the fear conditioning test. And just to give a general overview, because this task is a little bit more complicated than the other ones, the first initial day is training, and the mouse is placed in the fear conditioning box for a total of 5 minutes. Within those 5 minutes, there are 3 separate pairings of a 10-second tone followed by a very mild shock. So after the training day, we tested the mice 24 hours post-training as well as 2 weeks out post-training. During the actual testing days, the mice are placed back in the exact same fear conditioning box for 6 minutes. So the first half of the 6 minutes, there's no tone, no shock. The data points that we pull from this 3 minutes are just seeing how much the mouse freezes in response to being in the same context that they were previously shocked in. And the latter half of the 6 minutes, the same tone at the same volume that was played during the training period plays for the whole 3 minutes. And then we see how much the mouse responds to freezing in response to hearing that tone. So here are results for the 24 hours post-training. So you can see that we found a significant increase in percent freezing of the STZ group as compared to the buffers in both response to the context as well as response to the tone. In 2 weeks, we continued to find significant increase in freezing of the STZ group in response to the context, although we did not find significance in response to the tone. But you can see that it is trending towards significant as the STZ group has a greater percent freezing in response to the tone as compared to the buffers. So overall, our results in marble bearing, open field, and forced swim tests indicate that there's a heightened depressive-like phenotype in diabetic or hyperglycemic mice. And contrary to our hypothesis that the hyperglycemic mice would display poor cognitive performance, which would have been demonstrated as decreased freezing and fear conditioning, the hyperglycemic mice actually displayed increased freezing. And so in terms of the specific test, it's in the mice displayed improved. And after looking at current literature, this increased freezing behavior and fear conditioning could potentially be associated with an increased salience to aversive stimuli. And this could also be anxiety modulated. And overall, our lab is continuing to look into projects where we can explore the underlying mechanism of hyperglycemia associated depressive and cognitive outcomes. We would like to thank the Baylor Department of Psychology and Neuroscience, the Baylor P&I Lab, Dr. Vichaya, and our graduate student advisor. Thank you so much for your time. And are there any questions?
I had one question. Oh, I can put it in the chat.
Let's see, so I see the one question. So these were not rats, so we worked with mice, and they were the C57/J strain. That's the most widely used strain of mice to study human diseases. And there are multiple different strains, but these were not white mice, they were brown mice. And so there's different uses for different strains of mice, but those are the ones that we used. And so the other question about the anxiety task was relating to depressive symptoms. So there's not a strict, this is an anxiety task. The current literature usually associates this with, for example, the marble bearing, maybe if they have more anxiety, they might have engage in repetitive behaviors, but nothing is set in stone for these tasks. We're interpreting the results as they come, and current literature has also seen trends where marble bearing STZ mice might have decreased marble bearing. And so we just pull from the data that's coming in, and we are interpreting these results as this could potentially be relating to a lack of motivation, and that's why they're not bearing as many marbles. So it's not a strict, it has to be anxiety task, but we usually look at the current literature, look at the results, and interpret what we're getting in relation to what has been seen in current literature, if that makes sense.
Wonderful. Thank you guys so much. I also wanted to quickly let everyone know in the Zoom that our judge has joined us, Catherine Blandon. Thank you so much. But yes, we're going to move on to our last presenter, Esther. She is also from Baylor University. Thank you so much.
Okay, can everyone hear me okay? Okay, awesome. Let's see. All right. Hi, everyone. My name is Esther. I'm a junior neuroscience major, and I will be presenting on my research, which examines the efficacy of novel serotonin compounds in excessive voluntary alcohol drinking in male mice. So when we think of alcohol, we can generally picture two types of drinkers. So first we have your moderate drinkers. So moderate drinkers might enjoy a glass of wine on a special occasion or have a bottle of beer every night, but in general, moderate drinkers drink in moderation. And on the opposite end of that spectrum is your binge drinkers. So you can think of it as your typical college student. So for college students, a week might look like 5 days of studying, taking exams, and they might follow up the weekend with their definition of fun, which might include getting drunk, regretting it the next day, and probably repeating it again the next week. So binge drinkers have this pattern of drinking where they have excessive drinking days followed by abstinence and kind of repeating that cycle. So these are the two drinkers we have. And what I'm interested in understanding is their brain. I want to understand how the brain of a moderate drinker might differ from that of a binge drinker. So the broad research question that I ultimately hope to answer is what neurological mechanisms underlie these binge drinking behaviors as opposed to moderate drinking behavior. So before I jump into my project, I first want to highlight some statistics with you. So in the US, over 14.5 million people have alcohol use disorder, and alcohol costed the US at least $249 billion. 3/4 of that cost was due to binge drinking, and alcohol is also the third leading cause of preventable death in the US. And globally, 5.3% of all deaths are attributable to alcohol. So these numbers are quite staggering, and I think it does a great job highlighting the importance of alcohol research. But further, only 7.2% of alcohol use disorder patients received any sort of treatment, and less than 4% were prescribed FDA-approved medications. And currently, there are only 3 of them, and all 3 have also shown mixed results and moderate efficacy in their ability to treat alcohol use disorder. So that lack of effective pharmaceutical treatment is another aspect of alcohol use disorder that my project hopes to address. So without further ado, let's jump into my project. So my project utilized mice models of drinking. So I used C57 black 6-strain mice. They were all male. And I separated these mice into 2 groups with 2 different drinking schedules. So first we have your intermittent access group. So these mice had intermittent access to their 20% ethanol. What that means is, so I give them access to their ethanol on a Monday, then I would take away their ethanol on Tuesday and return it on Wednesday and allow them to drink, so on and so forth. And water was available at all times, so that adds an important voluntary aspect to their drinking behavior. And then our second group is our continuous access group. So as the name indicates, this group of mice had the privilege of having ethanol at all times. So all 7 days of the week, they could choose either between ethanol or water. So with these 2 groups, what I then did is I allowed them to drink for a total of 12 days. But I first want to highlight that kind of going back to my introduction, we talked about the 2 different types of drinkers. So our intermittent access would be your typical college student or like a binge drinker. And our continuous access was intended to model your moderate drinkers. So again, I allowed them to drink for a total of 12 days. After 12 days of drinking, I administered a compound called SB242084. Doesn't have a interesting name yet, but this is a serotonin 2C antagonist. So I administered the following doses written here. I also replicated this study with buspirone, which acts on a different receptor, the serotonin 1A receptor, as a partial agonist. But I will be focusing on the SB compound. So after administering the drug, I measured their ethanol consumption in the following 2, 4, and 24 hours. And I did also run some behavioral tests, specifically the open field test, to assess for any changes in locomotor activity or anxiolytic effects of the drug. Okay, so let's look at some results. So first, we're going to look at the SB compound group. So that's the serotonin 2C antagonist drug. So this graph is graphing the drinking behavior of the intermittent access group mice. Again, your binge drinkers. And what we essentially see here is that their drinking really escalates over time, over the 12 total days of drinking. And this is a graph of their ethanol preference. So ethanol preference is calculated by dividing the total ethanol intake by total fluid intake. So it's how much the mice prefer to drink ethanol over water. And interestingly, towards the end of the drinking schedule, what we see is that the mice almost only drank ethanol, which is a little concerning, but that's great for us because I think it really accurately modeled the kind of binge drinking behavior we might see in humans. So yeah, this was the intermittent access group. And when I administered the SB compound, what I found is that there was a dose-dependent reduction in their ethanol consumption. So what that essentially means is higher the dose of the drug, the greater the effect. The effect here being that the drug reduced their ethanol drinking. And we see this effect particularly for the first 2 and 4 hours time point, these first two graphs here. So around 24 hours later, the effect seems to wear off. So that's our intermittent access group. Now here we have our continuous access, and right off the bat, you can tell the continuous access drank a lot less than the intermittent access. Their ethanol preference also actually decreases over time. And when we put the two groups together side by side, the orange is the intermittent access group. And what we see is essentially the intermittent access drank a lot more. Their ethanol preference is higher. Interestingly, their total fluid was higher here, likely because they drank so much ethanol. So interesting thing about this project was when I measured the SB compound in the continuous access group, I did not find any significant effect of the drug, which is contrary to what we saw in the intermittent access group. So in the continuous access, the highest dose of 3.0 is not very different from the other doses or even the vehicle. So across all time points, we don't really see any effect of the drug. So that was really exciting to see. And I will touch on buspirone briefly. So this is the buspirone group. Again, the intermittent access group did drink a little more. Their ethanol preference was higher, and their total fluid consumption was a little different from the previous group. So for the intermittent access, administering the buspirone also did reduce their ethanol consumption, particularly for the highest dose of 10 milligrams per kilogram. The effect seems to wear off quickly after that time period though. And for the continuous access, it wasn't statistically significant, but we do seem to see some kind of trend in their ethanol consumption reduction as well. However, unfortunately, this data was significantly confounded by the fact that buspirone had significant sedative effects. So this is a graph of the data from the open field test. And what it's essentially showing is that when I administered the highest dose of buspirone in the mice, it led to them having really high immobility compared to the vehicle group. So vehicle group is the mice that received a saline solution. So essentially, these mice did not move at all. So reduced locomotor activity would probably have confounded their drinking behavior. So yeah, that was unfortunate, but yeah, what do these results mean for us though? So there are a couple of implications from my preliminary study. So first is that binge-like drinking behavior and moderate drinking behavior may have different underlying mechanisms. Specifically from our results with the serotonin 2C receptor. So we can conclude that the serotonin 2C receptor may be involved in mediating binge-like drinking behavior, but maybe not so much for moderate drinking behavior. And another important implication based on that is that perhaps future research may benefit from targeting this receptor when developing new treatment options, or maybe the SB compound itself should be considered a candidate as a potential novel alcohol use disorder treatment. So moving forward, aside from adding more mice to my current study to increase my power, I'm also really excited to look at sex differences, which I think I'll be working on this summer. So my current study only used male mice, and literature has actually shown there's significant sex differences in alcohol use disorder in humans. There's also sex differences in rodents' drinking behavior. Furthermore, women are actually more vulnerable to the health risks associated with alcohol. Unfortunately, a lot of research in the alcohol field has only focused on males, as it is with many other studies. So there is this large gap in our understanding of alcohol use disorder in males versus females. So I'm really excited to see if there's any sex differences by basically replicating this study by using female mice. And another fun neuroscience aspect I could add to my project is looking at serotonin expression in different areas of the brain, comparing how the serotonin expression might differ between the intermittent axis versus continuous axis through methods such as immunohistochemistry. But yeah, these two are the main future directions I'm really excited for. And that's pretty much it for my project. So I just want to end by thanking my Hua lab. I'm giving a huge shout out to my PI, Dr. Hua, for allowing me this opportunity to research with her. And of course, all of you guys for listening to my presentation today. Thank you.
Thank you so much, Esther. Does anyone have any questions? Any at all? You can ask them in the chat or just unmute yourself.
I have a quick question about the compounds that you decided to use. I know buspirone is like an anxiolytic drug or anxiolytic drug. Is there a reason that you chose that one? Like, is it one of the FDA approved ones for alcohol use disorder, or did you just like think that maybe it would be like implicated?
Yeah, so all the drugs we used, I used in my project, they're not FDA approved for alcohol use disorder, but I was interested in looking at how different serotonin receptors are involved. So I was trying to look for drugs that bind or act on different receptors. So yeah, the SV compound was the 2C receptor, which I used that one because there were previous studies that have also implicated it, decreasing alcohol consumption in rats. And for buspirone, there were some studies like from way back, but it wasn't really explored in recent years. So I thought it was, worth looking at, but I had no idea it was a sedative. So yeah.
Yeah, I was just curious about it because in my class, I know it's approved for anxiety, but doesn't work very well for that either. So. Thank you.
Any other questions? Oh, sorry.
Esther, did you apply both drugs to both groups, the moderate drinking and the continuous?
Yes, yeah, I did.
Did you have to account for one versus the other in your analysis at all?
Sorry, what do you mean one versus the other?
So they're both antagonists, correct?
No, so SV compound is the 2C receptor antagonist and buspirone is a 1A receptor agonist. Yeah.
Okay.
All right, if there's no more questions, I'll be doing a quick photo with everyone if that is okay. Thank you to all of our presenters and thank you to our judge. But yes, we will take a quick photo and then y'all can hop on the grad Zoom, the grad link Zoom at around 11:15. But yes, thank you to everyone. All right, you guys want to turn on your cameras? It'd be great. I saw some of you did that, but if all of you do, that'd be wonderful. Okay. Ready? Wonderful. Okay, thank you guys so much. If the judges could stay on, presenters can stay on too if you'd like to debrief it all, but the rest of you just hop on the grad Zoom at around 11:15. But thank you for coming. Thank you to our presenters.